My name is Tom Walton. I'm the Chief Business Officer for Carisma Therapeutics. I'm delighted to be here today with everyone in Rome, and thank you to ARM for the opportunity to participate in the panel earlier. Hopefully some of you managed to catch on solid tumors, and to tell you more now about Carisma and our engineered macrophage platform. Well, we're figuring that out. I'll tell you a bit more about the company. We're based in Philadelphia, and we spun out from the University of Pennsylvania, the Center for Cellular Immunotherapy, and that's the group led by Carl June. The company was founded in 2016. To date, we've raised just over 300 million, a mix of private, public finance, and a significant partnership that we signed a couple of years ago with Moderna Therapeutics. There we go. Before we go into the presentation, just draw your attention to our forward-looking statements. So I touched on earlier some of the key challenges in the solid tumor space. I'm not going to go over those again. I would just say that the initial focus of our technology is on CAR macrophages for solid tumors. Our lead program, which we're in the clinic with, is a CAR macrophage targeting HER2-positive solid tumors. We recently announced a slight change in strategy for that program, where we are basically winding down the initial program, which was a CAR macrophage, and focusing our resources on the CAR monocyte product. This has the same CAR, it's just in the precursor cell in the monocyte, and that has a number of advantages that I'll talk about as we go through the presentation. In addition to that HER2 program, we have this significant partnership with Moderna. The focus of that is in vivo oncology, so using Moderna's LNP mRNA platform, our macrophage engineering capabilities to deliver CARs to macrophages and monocytes in situ within the body. And then finally, we recently announced our first non-oncology program. This is a liver fibrosis program, which we're very excited about. And again, I'll tell you more about that as we go through. In terms of our corporate cash position, we have cash on hand to last through into the third quarter of 2025. We have what I think makes us unique, a very strong IP position, where we have dominant patents on CAR macrophages and CAR monocyte that are irrespective of target, irrespective of starting cell. And then potential opportunities for partnerships. We see this as a platform which is not just limited to solid tumors, liver fibrosis, but where we can go beyond this into other diseases, autoimmune, neurodegeneration. As I mentioned, the three key pillars to our current strategy are the HER2 clinical program. Our goal there is to identify by 2025 the regimen that we can take through to commercialization. In 2024, we have recently announced the IND clearance and initiation of the first ever CAR monocyte study targeting a solid tumor. First cohort of that study is a 3 billion cell dose of the CAR monocyte. Then we'll initiate the second leg, which is a 10 billion cell dose, a significant increase over what we were able to deliver with the CAR macrophage program. The second leg of the stool is the collaboration with Moderna for in vivo oncology. This has the potential for essentially an off-the-shelf therapeutic, which is personalized to the patient, which is generating the product, the CAR macrophage, in situ within the patient's own body. We recently announced lead selection for the first program. We have another 4 programs that we're already working on in preclinical development. We don't have guidance for 2025. Moderna are quite cautious, I guess, in terms of putting out future guidance, but we plan to move this program forward rapidly towards the clinic. And lastly, the liver fibrosis program that we've been working on for just over 18 months. We think there's a very nice story here, a nice application of our technology in late-stage liver fibrosis patients. We will be sharing data shortly at an upcoming conference with the initial proof of concept on the initial construct, and the goal is to move this rapidly forward through IND-enabling studies. So focusing in a little bit more detail on the HER2 program, We shared data at the end of last year with the initial product, as I said, the CAR macrophage, that was the CT0508 product. What we were able to show there in this initial phase 1 safety study in very heavily pre-treated HER2 patients was great feasibility. We were able to make large quantities, high quality cells, good CAR expression, good potency. The cell product has so far been very well tolerated, very safe. We've seen no dose-limiting toxicities, no severe CRS. Some instances of grade 1, grade 2 CRS that were transient and rapidly resolved with Tylenol, so nothing that's really given us cause for concern. And we were also able to see the mechanism of these cells play out. So macrophages have a unique triple mechanism, which we think will make them particularly effective in solid tumors. They traffic actively into the tumor. They're able to modulate the tumor microenvironment, and then they are a professional antigen presenting cell. And essentially in this first ever in man phase 1 study, we were able to see all aspects of that mechanism play out. As a result of that, we did see some impact on the patient progression. We saw stable disease in a number of these patients, notably all 3+ patients, suggesting the effect is independent. But clearly we need to go one better. And our plan moving forward is to address what we saw as the two key, shortcomings with the initial CAR macrophage study. The first of those is that even though we're able to deliver high quality, large numbers of the macrophage cells, the effect of the product is also dependent on having a reasonably robust T cell system. In the majority of these patients, they really came into the study in a very beat up way. Some of them 10th, 11th line, really with no T cell response at all. So we think the addition of a T cell checkpoint inhibitor is going to be critical in this late stage patient population. So the first stage of that will be the combination of the macrophage with pembrolizumab that we started last year and will read out in the second quarter of this year. The second part of the strategy is to increase the dose. So if the product appears to be working, it's doing what it's supposed to do, and it's safe, drug development 101, where you ramp up the dose. With macrophages, it's tough to do that. These are a non-expanding cell population, so the only way we can do that is to move to the monocyte, which is the precursor cell. With the monocyte, we can manufacture approximately 5 times the dose of cells, so we can significantly increase the dose. This is the CT0525, so the CAR monocyte product. It's a pretty standard CAR. It's actually the same CAR we used on the macrophage. CD8 hinge, transmembrane, CD3 zeta, CAR T signaling domain. It's an autologous product again. We use our Ad5F35 vector to engineer the product. It has the advantage that it, as well as delivering the CAR, it also polarizes the cell into an M1 pro-inflammatory phenotype. In terms of advantages of the monocyte over the macrophage, we've talked about the increased cell number. We can get 5x the number of these cells from a single apheresis. The cells also traffic significantly better. Macrophages are large, sticky cells. They tend to get stuck in the liver and in the lungs, and we think this is essentially what's been happening in this initial phase 1. We have located the cells in the tumor microenvironment, but in very low numbers. So we think moving to the monocyte, the traffic's significantly better. We'll get more of the cells in there, obviously a higher dose. And then what we've also seen is that these cells stick around for longer, around to 10 times the lifespan, and then they're actually more potent. One of the reasons we chose the macrophage initially was we weren't sure if the monocytes would transform into macrophages within the TME. What we've seen now preclinically is not only do they differentiate into macrophages, but they're actually slightly more potent. They're more effective at phagocytosis. They are better antigen presenting cells. So we think the combination of all of this, coupled with the fact that the CAR monocyte, the manufacturing process is far more rapid, it's a single-day manufacturing process, makes this really the ideal product to move forward. It's just some of the preclinical data on the characteristics of the monocyte. You mentioned the higher cell dose, 40 times the tumor infiltration, 10 times the persistence. If you put all of this together, you end up with around 2,000-fold increased exposure of the product within the tumor microenvironment, which we Obviously, we don't know till we go into the clinic, but we feel confident will be enough to move some of these stable disease patients to being responders, and that's our goal in the next phase of the clinical study. This is the trial design and the process. Essentially, we're targeting the same patient population, 2+ and 3+ HER2 patients. The process is exactly the same as the macrophage, only we don't differentiate the monocytes into macrophages. We treat the patients with GCSF to mobilize them. There's no lymphodepletion, unlike T and NK approaches. We don't lymphodeplete, they don't have chemotherapy. We apheresis the PBMCs, select out CD14 positive monocytes. In this case, we're not differentiating to the macrophages, we just leave them as monocytes, add our vector. And then what we've seen is we can generate on average from healthy donors anywhere between 10 up to 15 billion cells from a single apheresis. We think it's likely we'll get somewhere in the order of 10 to 12 billion cells with the monocyte product. The first cohort will be up to 3 billion cells, and then we will move to up to 10 billion with the second cohort. There is a potential here, depending on how many cells we generate, that we could potentially look at redosing in the future as well. Primary outcomes, obviously it's a phase 1 study, heavily pretreated population, but we'll be looking at safety, tolerability, manufacturing feasibility, and then also a range of correlative studies as we've done with the macrophage, and ultimately looking at patient impact and responses. Talk a little bit more about the collaboration with Moderna. This is a deal that we signed 2 years ago. We received $80 million up front for up to 12 targets, all in solid tumors. Potentially $3 billion of milestones if all of those programs are successful. And critically, the whole of the program is fully funded by Moderna, so they pay for everything that we do with them. We have a team of about 25 scientists who are purely dedicated to the Moderna investment. The opportunity here, which we see, is essentially to be able to take what we've learned with the ex vivo autologous product and now using Moderna's platform. Obviously, they're the world leader in LNP mRNA, had huge success with their vaccine platform. They've shown some very, I think, exciting data with their oncology vaccine program as well. But to be able to take that and to be able to re-engineer the cells actually within the patient's body, so in situ, essentially taking an off-the-shelf therapeutic and developing a purely personalized cell therapy within the patient. We shared some initial data from this at SITC last year, created some interest. This is one of the studies that we shared there in a metastatic pancreatic cancer model. What we're able to show with this off-the-shelf treatment approach essentially was effective tumor control in a number of models. The LNP is myeloid tropic, so it ends up trafficking effectively into the myeloid compartment. It gets basically gobbled up by the circulating monocytes and the tumor-associated macrophages. That delivers the CAR into the monocytes and macrophages. And then we were able to see, again, the whole mechanism of action of those monocytes, macrophages trafficking into the tumor, warming up the tumor, recruiting in other effector cells, T cells, NK cells, and launching an attack directly against the tumor, and as we've seen here, managing to elicit effective tumor control. Coming up to time, so I'll just talk very briefly about our liver fibrosis program. This is our first non-oncology program. If you heard the presentation earlier, one of the discussion points we had was, why use cell therapy? How do you select indications? So when we started to talk about moving outside of oncology, what we wanted to do was to really focus on an indication with A, huge unmet need, and with B, where there was a really strong rationale for using an engineered macrophage. Liver fibrosis, macrophages, or Kupffer cells as they are resident in the liver, play a critical role in the pathogenesis of this disease. It's our belief that by re-engineering them to be anti-inflammatory, engineering them to be anti-fibrotic, and lastly, pro-regenerative, we can go one better than just simply deploying wild-type macrophages, where there's been some, I think, fairly encouraging clinical data already shared from a group out. As you see here, this is a drug-induced model of liver fibrosis. The non-engineered macrophages were able to effectively reduce the fibrosis by around 50%. But when we engineer these cells, and we haven't disclosed yet what the payload is, but we're looking at a number of different constructs, we're able to completely remove the fibrosis from these animal models. At the same time, we can reduce the inflammation, drive regeneration. So in one product, you can essentially address all of the aspects of the pathology. So to close, Carisma Therapeutics is pioneering the development of this new class of cell therapy. We have 3 key areas of focus: ex vivo oncology, in vivo oncology, and now liver fibrosis. Very excited about sharing additional data as we go through this year with you. I think I'm at time, so please give me a shout if you have any questions after the session. Thank you.